This episode is presented by Ashton Thomas Private Wealth, guiding families and institutions with clarity today and strength that endures for generations.
Evolution is not always doing what we want. It's optimizing some reproductive function is sometimes a little bit obscure in the way that we think about it today. There's no law of physics that says you can't live longer than a bowhead whale. It's all negotiable, we think. We'll see.
Aging is evolving. No longer are we subject to forces beyond our understanding and control. We have charted the landscape and explored the frontiers of aging.
What was science fiction is close to becoming reality, restoring sight, repairing tissues, reviving cells, organs, and maybe even our minds.
I'm Eric Verdi, CEO of the Buck Institute.
And I'm Brianna Stubbs, a scientist here at the Buck.
On this podcast, we dive deep into geroscience, studying the intersection of aging and disease with some of the brightest scientific stars on the planet.
Join us because
we're not getting any younger, yet. Hi, Brianna.
How's it going, Eric?
It's going well. I just sat down with George Church.
George Church?
Yeah,
Huh?
the George Church.
Gosh, he's a giant in the aging field. It's impossible to be in this field and not heard of George Church.
He is, and he's one of the most provocative thinkers and doers in the field. And,
What makes you say that? Provocative?
well, his science is always big ideas, like when he's proposing to resurrect the woolly mammoth.
Oh, right.
That's one of his
I
projects.
kind of like to see that. Really? Huh.
So, yeah, and he has the same ambition in terms of aging science. He wants to really stop aging, and he's going through a track which is very few people are going to, which is gene therapy.
Kind of a lot of people would have a bit of like the heebie-jeebies with the gene therapy. It's definitely a controversial space, not easy to be a pioneer, and you have to think a certain way, be a certain type of person to just sort of, not care what other people think if you're going to go hard into that space.
And that sort of epitomizes him. He doesn't care what other people think in terms of, should we do this, should we not? He's going to do it first in animal models and then hopefully next in humans.
Wow. Well, I'll look forward to hearing your conversation. It sounds like it's set to be a fantastic one.
It's an amazing conversation. Check it out. I'm delighted today to welcome George Church to our podcast. We're not getting any younger yet, but with people like George on board in the field, I think it is only a matter of time. So welcome, George. Delighted to have you here.
I'm delighted to be with you today.
Your career has been truly expansive and actually has affected many of the people in our audience and in the world in ways that they might not even suspect. So just to give it a little bit of background, when I started sequencing 40 years ago, it would take us 3 days to analyze 400 base pairs. Today, we can sequence 3 billion base pairs in a few hours. And the first human genome cost $3 billion to sequence. Today, we can do it for about $150. And in many ways, next-gen sequencing was the basis for many of these discoveries. But it's not only, in terms of human genome sequencing now, next-gen sequencing is part of many of the experiments that we do at all levels. So really amazing contribution. And to have you in the field of aging research really is exciting. So how did you get interested in aging?
Yes, well, I think all of us have had a cherished family member die of old age. It's going to kill 90% of us, at least in the industrialized nations. And but the real turning point was when a postdoctoral fellow came to my lab, Pedro de Magalis, who is still a mover and shaker in the aging field. He and I embarked on some studies of zoological components, comparing different body masses and longevities and of various animals, including extreme outliers. When you basically plot, and these would be extremely long-lived relative to their cousins, other closely related animals. And these were the bowhead whale and the capuchin monkey and naked mole rats. And we started a couple of databases, which are still, I think, some of the main databases that are used for human and animal aging and age-related genes. And we used those genes very soon thereafter to test out dozens of genes, about 48 genes at once in gene therapy. So that was my start.
Yeah, so that's actually really interesting because one of those papers on the bowhead whale discussed something that is not really discussed much in the literature. It should be a lot more discussed. It's the whole idea of scaling, of aging as a function of body mass. How does body mass translate into shorter or longer lifespan?
There's a correlation that as you go up in body mass, you get longer lived animals. And in fact, many of the large body mass, long-lived animals have extraordinarily low cancer rates, which is kind of contrary to what you would expect, because cancer is a disease that happens as you replicate your cells. The more times you replicate, the bigger your body, but also the more errors that you could have. And all it takes is one or two errors to start a process of cancer formation. So it's very unusual that things like whales and elephants have such low cancer, while things like mice, which are really tiny, have high. And then as you have this nice plot of where you go up with body mass and longevity, there are a few outliers that are even more long-lived than you would expect from their body mass, and those are the ones I listed were good examples of that. Humans are actually a little bit above the line as well, meaning a little bit longer lived than you would expect. Yeah, so I think that's what you were looking for.
This is one that is still not fully understood, but and underlies the whole field of longevity. And the concept of longevity quotient is also, how does your lifespan correspond to what your body mass is? And it turns out that humans are in some way doing extremely well already. That is, for our body mass, we're living longer than we would expect. And mice do exactly the opposite. And for me, it's been one of the arguments why it might not be so easy as we hope to actually increase lifespan dramatically in humans, because in some way I've argued that maybe we are already evolutionary optimized by virtue of being predators and apex predators.
Well, I don't doubt that we are somewhat optimized. And the reason we were studying these long-lived animals is that there might be multiple ways that they use to do this optimization. And if you combine them, you might go beyond. Evolution is not always doing what we want. It's optimizing some reproductive function is sometimes a little bit obscure or frugal in kind of a penny-wise, pound-foolish way in the way that we think about it today. Because today we have no shortage of food. So we have in a certain sense, no shortage of body energy to repair, to do like double checking of processes to make sure everything's going okay. And that was too expensive for our ancestors in a kind of a metaphoric evolutionary sense. But now we have a new set of priorities. So I think what we're really doing is collecting a set of genes, proteins that are encoded by those genes, and seeing what combination of them can push things out. There's no law of physics that says you can't live longer than a bowhead whale. It's all negotiable, we think. We'll see.
George, there are two sort of broad schools, and they're not necessarily exclusive of one another in the aging field. One that is focused on repairing the damage that has been done, in some way the replacement school. And the other school is that we should actually prevent the damage from occurring. What do you see is more important, and how do we tackle both of those directions in the field?
Damage can include damage to DNA and proteins and so-called epigenetics, which is how the DNA proteins associate with one another and are produced. Yes, I think our ancestors we're very frugal. We could spend more energy on increasing our rate of repair and so forth. It's actually quite good, but we can make it better. We could put in redundancy. This has been done in animals. You can put in redundant copies of genes that once they get damaged, they lead to cancer. Now that we have ability to make a variety of cell types from stem cells from the patient or possibly generic stem cells, these are distinguished from most of the stem cell therapies in the world, which have not been properly vetted, have not been through FDA approval and so forth. But future stem cell projects, or where we can make the precursors to various cells and do replacement, that might end up being better than just trying. I think we can do both. We can increase the repair processes preemptively, and we get via better enzymes inside of the cells, but we can also replace the cells. Even in the brain, even though the brain doesn't do a lot of that, it can be coaxed into doing more than it normally does. So I think these are both very promising pathways, and it's not quite clear the ones I've described which path they're working. We are working on one other thing that didn't mention that is appropriate here in terms of making tissues and organ replacements. We are making organs both based on human cells and based on reprogramming pigs. The pigs are much further along. In fact, we have someone who still has a very active pig kidney after now 4 going on 5 months. I met with him the other day, and he's just having a great time relative to kidney dialysis. So, but this could be much more general than fixing really broken organs. This could be something where it's done preventatively or curatively body-wide.
Truly exciting, and the whole idea of reprogramming for aging has taken the field by storm. And I just want to spend a little more time on this for those members of our audience who haven't heard about this. So maybe let's step back to Shinya Yamanaka discovered these 4 factors that by themselves are able to take any somatic cell, by somatic we mean a cell from the body, and to reprogram them into pluripotent embryonic stem cell, meaning a cell that has the ability to eventually to divide forever and to become any specific tissue. And so this created a revolution. Do you want to take us to the next step of what Ipsisua Belmonte did in terms of the partial reprogramming and how this led to this work?
Well, before we get to Belmonte, the observation, which wasn't really intended to have anything to do with longevity as far as I know, or at least the early adopters were mainly looking for stem cells for which they could reprogram. First, they reprogram it from an adult cell into an embryo-like cell and then back out to a different adult cell. That was the plan. So you can make tissues like to solve diabetes and so forth. The goal was to make a new, have an infinite supply of your favorite missing tissue. But but the observation was that it basically reverted it from, you could take it from an 80-year-old, and with treatment with these 4 factors, you could get to essentially an embryonic state. And by partial reprogramming, the Belmonte experiment is you could get something that's just rejuvenated without being dedifferentiated. And that's important because the totally dedifferentiated one, firstly, doesn't do its function anymore. It doesn't do a mature function like producing blood cells or skin cells or whatever. It's pluripotent, meaning it can do almost anything, but it isn't doing anything at that moment. So that's too far. So partial reprogramming was hoped to and was shown to just cause rejuvenation without complete dedifferentiation to an embryonic state. So it stays in its mature state. And but those experiments were not really done in a format where you could call it a gene therapy and take it to the FDA, and that's what we added to it. And again, so we weren't going for longevity, we were going for reversing particular age-related diseases, but we decided to let those animals keep living and just see how far they went. And sure enough, there was a longevity effect as well.
Yeah, and as we discussed earlier, this has taken the field by storm, and not only the field, but also the public imagination, because these were and these are some of the earliest instances in which we can truly talk about rejuvenation, sort of rewinding the clock back. And
this is very clearly aging reversal at a cellular level.
We know that the field has embraced this in a huge way. About a dozen companies here in the Bay Area, all targeting rejuvenation using the same type of approach. One of the key questions, we can clearly rejuvenate in vitro. We can also partially rejuvenate in vivo, in animal model. One of the major barriers to bringing this to the clinic has always been the idea of the vector. How do you, if you're going to be transducing this into a human, hopefully you would have to do it in most cells or most organs, or at least in a significant proportion. So can you tell us a little bit of how do you see us addressing this barrier to, having the right vectors, for example, go into, because you could rejuvenate the whole body minus the brain. This wouldn't help us. And so the brain still remains a tough place to go in terms of, getting enough cells transduced. What's your thinking in terms of where's the field moving in this direction? Do you really think it's going to be doable to get global reprogramming?
Well, there's two directions that it could go that I think are quite hopeful. One is that the first three factors we talked about, which were aimed at the blood, or even the Yamanaka factors, which you at first blush you think they're not secreted into the blood, they are kept in the nucleus. They're transcription factors. They work on the DNA, and so whatever cell, in order to have an effect on all your tissues, you would think you would have to get it into each of the tissues. In other words, the AAV that you maybe inject into the blood would then have to go through the blood and cross the endothelial layer, that's the inside of the blood vessels, into each tissue, and then find its target and do its thing. Gene therapy in general is not particularly efficient for any tissue other than the liver. So solution number one is that maybe the Yamanaka factors can act the same way that our soluble factors. We intentionally focused on soluble factors originally, but we felt we had to do the Yamanaka factors as well. And maybe they work the same way. They rejuvenate the liver, and then the liver produces who knows what, but it goes through the blood, and the blood goes everywhere. And proteins, even though it's hard to get gene therapies delivered everywhere so far, and that'll be number two when we get to it, is the proteins are much more effectively spread throughout the blood. And many proteins cross the blood-brain barrier and get into the brain, which was your favorite tissue a few seconds ago. So number two, though, is to actually target those cells, and we do to not count on the blood carrying a protein to those, but actually having the virus itself go to every tissue, like the brain. And the brain is not just your favorite tissue, it's everybody's favorite tissue, because we have an epidemic of cognitive decline, a lot of it caused by Alzheimer's and related diseases. And if you're going to live past 90, you got a very, very high chance. I mean, it's almost guaranteed that you're going to have some cognitive decline. So anyway, one of my other companies, not Rejuvenate Bio, but Dino Therapeutics, set this as their mission was to get AAV that normally, and in fact, most viruses and most non-viral deliveries go to the liver. They said, let's get it away from the liver and towards the brain. And to do this, there were two key tools that had been used separately with some success, but together they're amazing. And those two tools are artificial intelligence, machine learning, and the second one is large libraries. So use artificial intelligence to make large libraries of variations on the capsid that is going to be targeting the brain, but normally it doesn't. Change the surface so it will recognize various components that lead it to the neurons in the brain. And the AI helps you make sure that you maximize the bang for the buck for the number of mutants. So we're going to make about 1 million mutants, or we did make 1 million mutants. And we want to eliminate those that are clearly deleterious based on AI, and those that are clearly redundant and neutral of no interesting change, and focus on lots of change that isn't going to kill the capsid. Oh, and then the third trick is to inject it into an animal, which is the best human model we have. It's normally avoided because they're very expensive, which is primate models, baboons and rhesus monkeys and so forth. But the reason we could afford it, doing 1 million primates would be unaffordable to almost anybody. We did 1 million at once, so we can inject all these payloads where the DNA encoded a barcode and the capsid was different for each virus, and then we could find the ones we could just take a little bit of brain, a little bit of liver, and say, detarget the liver and increase the target of the brain. And we ended, after that process, we ended up with 100-fold improvement in delivery to the brain and a 10-fold detargeting of the liver. And I think this is a general thing we are hoping to do this with every tissue that is of significance to age-related diseases. So definitely the brain and the immune system, very important targets for aging.
Agreed. There's a recent number of papers suggesting that those two organs, brain and immune system, which are the only sort of distributed organs, actually play a rate-limiting role in aging. That is, if you have immune aging, you get organismal aging, the same with the brain. So I think it's fascinating. I mean, immune system is much easier to target, but it's amazing to hear that using these types of selection techniques, you're able to enrich for new AAV vectors that will bring the payload to the brain. It's fascinating to see. So we're on the verge of a revolution. In some way, the revolution is already ongoing. It started about 20 years ago by the work of Gary Rifkin and Cynthia Kenyon and Tom Johnson and all those, showing that aging was actually tractable using the tools of molecular biology. And we know these tools have been incredibly powerful for any subject that we've tried to tackle. So where do you see this field going?
Yeah, well, I think one thing that seems to be happening is that clinical trials are getting shorter, safer. So it used to be that long trials were necessary to get safety. I think we're getting safer. This includes gene therapy. So the COVID-19 vaccine was delivered basically as a gene therapy. It was a lipid nanoparticle outside and a messenger RNA inside. And there were some of the vaccines that were actually viral outside and double-strand DNA inside. And those went through about 6 billion people received those. And so they were safe and effective. And that was about, some of those were developed in 11 months. So that was much faster than the normal of 10. And we just saw a gene therapy developed for a single individual baby. There was a kind of a hurry to do that quickly because the baby had a unique disease which was not on the list. That's like the ultimate in individualized therapy, and that also went quickly in months, and probably could be repeated for additional diseases. Now, I think the real payoff for gene therapy is for very common diseases, because there it's affordable. So it's like $20 a dose for the COVID vaccines, and it probably could be in that range for anything that has a really big market of billions of people. and certainly we're all susceptible to aging, and the consequences of aging are not just death, but ability to do one's job. So huge economic consequences.
What you're saying is that you see within the next 10 to 20 years, gene therapy based on these vectors and this technology is going to become much more available and allow us to do much more customizable type of treatment. So that's your vision.
Yeah.
What is your whole philosophy about aging in general?
Well, I don't think it's that fundamentally different from most aging researchers who are studying aging. Well, maybe it's a little different in that I think that we need to take a lot of shots on goal, but at a very close to human level. So I think it's not sufficient to get things to work in tissue culture cells. We need at least human organoids, and maybe humanized primates would be even better, because a lot of these are systems problems. They involve their interactions between the immune system starts to fail, and then neuroendocrine and kidney failure and so forth. You have multiple systems failures, and so you want to see how that works out in an intact body. So I think we're going to start the kind of multiplexing that I was talking about, where you could inject 1 million different capsids just to see where they would go, is just a start. You can also not only see where it goes, but the kinetics at which it decays, how long it lasts, and whether it solves a problem, at least locally, and so you can sample each tissue and see whether the drug, in this case, the gene therapy, is being effective. And the nice thing about gene therapies, in addition to all the advantages I've already mentioned, is they contain barcode tags. And so if you can see that it's doing something good here, then you can check out the barcode tag and know which of the million viruses was doing the trick. So I think that's going to be increasingly the case. And I think, moreover, we're getting to the point where we can try various combinations. Combination therapy is very common in certain parts of pharmacology, in particular cancer, fungal, bacterial, and viral pathogens. In each of these cases, you use 3, 4, 5 different drugs that hit different targets, or maybe some of them hit the same target, to make sure there's no escape. Here, we're not using it because we're worried about escaping so much as there are multiple pathways we have to get simultaneously. Otherwise, it's like whack-a-mole. We knock down cancer, but then 10 other things will kill you. And so you want to get all 10 at once. And you get close enough to that, we might hit the point where science is advancing faster than our aging is.
George, I think a lot of your work is bringing us to the edge of a whole new field, which is if you combine, deep sequencing, genome sequence analysis, CRISPR-induced gene modification. This is bringing us to the edge of an area where we could take control of our own destiny in some way. Introduce mutations in humans, correct mutations. I mean, this is what we're doing first. We're correcting mutations if a kid is sick. But the same technology, in principle, could be applied to actually improving our own lot. And so this brings up a whole series of questions. And I think in many ways, I think humanity is not ready for this. But I would love to hear, as we saw by an attempt that was done in China, I believe, to make children resistant to HIV by knocking off the receptor. So what is your thinking about the ethics of us in some way taking control of our own destiny via genetic manipulation in the future?
Well, first of all, I think what you're talking about is not taking control of our destiny, it's taking control of the destiny of our descendants. And I would, my critique of what's been done so far and what we've been considering is that there are two big problems. One is that, let's say you want to solve, one of the major health threats today, which is cognitive decline. Typically, it's late onset after age 80 or 90. And so the clinical trial for that would, if you wanted to do it through the germline, through babies, would be 80 years. And if it fails, then it's another 80 years. So we're talking about a century and a half from now. And that's just not feasible with current economics and health economics. The second one, and a big one, is that there are 8 billion people on the planet who are paying all the bills and would love to benefit from this as well. And they are past the point where this would be applicable. This would only be applicable in germline. So I think those two things are real showstoppers, in addition to all the ethical qualms, which are a little bit vaguer as to and a lot of ethical things change from location to location on their planet and time and age to age. For example, there was near consensus in the United States against interracial marriage and in various other parts of the world, and these things change. But in very practical terms, those two things of the length of the clinical trial, and the inapplicability to most people, all people that are currently born. But the answer is we can enhance, augment adults, and that's going to be more ethical. It's going to probably be more affordable because you can develop something that's useful for 8 billion people rather than a small subset of rich babies, and that means that the cost might be in the same range as a vaccine. And we are already enhanced in a certain sense. Vaccines are an enhancement relative to our ancestors, and they're increasingly being done by something like gene therapy. So I think that we'll develop vaccines against cancer in a certain sense, metaphorically, vaccines against a variety of age-related diseases, hopefully all of them.
Well, that's fascinating. One that obviously will have to be, again, surrounded by careful ethical consideration, because you could also imagine, is it going to be, what happens to neurodivergent individuals? Where do we lay down the line in terms of what should be improved, what should not be improved, and so on. So it's going to be an interesting time. Do you think our governments are ready to tackle all of the ethical questions that will be coming with these possibilities?
I would like to believe that we, the people, and the governments will be ready for it. I think neuroatypicals, I would consider myself among those, being narcoleptic and OCD and dyslexic. They should at least be able to have some choice in the matter, right? So in other words, for example, I'm not in any rush to cure any of my problems. If I fall asleep without warning, which means I can't have any job that involves driving or heavy machinery. But there might be some that if you could, some brain dysfunction you can't solve because your brain is so messed up by say microcephaly or something like that by the time you're born. But many of them, you might be able to dial them up and down. And so, you could have a phase where you're incredibly focused on something and you're actually not very capable of social interactions, and then you dial it back and you're the opposite. And so I think that's one way you can thread this needle a little bit. But I agree with you, it has to be very carefully done, and it goes beyond just safety and efficacy. How do we make sure that it's not addictive, that it's not, some way that you could abuse it where you could, slip it into somebody's drink. So we have plenty of neuroactive compounds that are abused both by the individual and by other individuals inflicting them. So, and governments are not entirely innocent as well. Making super soldiers would be something that we would have to be very cognizant of. It's even possible there would be, just like there's a race to make AGI, artificial general intelligence, there may be a race to make natural general intelligence. That is to say, enhancing humans. And those races are not, they're not races against, where we're trying to race against some infectious agent. We're just racing for the sake of racing, and I think that's very dangerous.
No, I agree. Thank you. That fascinating way to look at the world. In some way, when people ask, about some of these questions, I tell them, look at the fraction of the population that's drinking caffeine. It is a neuromodulator, a very powerful one that many of us use with great success. So I think there are many other ways that the same paradigm could be implemented. To finish, I think our audience always likes to hear what people who study aging do for their own aging. Is there something that you can share with our audience in terms of what do you focus on? I mean, we all realize that some amazing treatments might come in the future, and I tell people, do everything you can now for the next 10 to 20 years to stay in as good a shape so that you can benefit from it. So what is it for you?
I'm not a big fan of anecdotes. Plural of anecdotes is not data. I am very much a fan of volunteering for clinical trials. And I've been a guinea pig most of my life in one way or another. IRB approved studies, meaning human subjects, researchers, proper ethics. I discourage people from doing things that haven't been tested. And if they are going to do something that hasn't been tested, it should be in the context of one of these clinical trials. Especially, they have to be a control group. Now, nobody wants to be in the control group. And I think one of the answers to that is what's called a crossover trial, where you blind, you either have the drug or the placebo or something like a placebo that's like a previously approved drug, and then halfway through, the ones that were on the placebo become the experiment and vice versa, and the patients or the research subjects don't know which it is. And so that means that everybody at some point or another is not in the placebo group, which is where they want to be. And I think that's the point, is that we will need lots of volunteers to get a lot of these things tested.
I've argued that our field will be defined by the rigor and the method that it uses, and even though the FDA is quite often criticized for sort of being in the way of the aging field, I think the FDA has been there to protect us and has led us to where we are today. And I think we should recognize this. So yeah. I love the idea that you embrace this idea of rigorous clinical trials to demonstrate the safety and the efficacy of what we're going to be doing in the future. So on this, I want to thank you for sharing your insight with us, and look forward to seeing you in the future.
Thank you. It's been a pleasure talking to you.
Likewise. Thank you.
Thank you so much for listening. Please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts.
We're Not Getting Any Younger Yet is produced by Vital Mind Media. The Buck Institute's very own Robin Snyder is the executive producer. Wellington Bowler is right next to us here directing the recordings, and the esteemed Sharif Azad weaves the show together for you.
If you're listening to this podcast, you know that there has never been a more exciting time in research on aging. Discoveries in the labs are moving into the clinic to help us all live better longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to learn more and to donate. This episode was sponsored by Ashton Thomas Private Wealth, where discipline shapes vision and vision builds legacy. Learn more at ashtonthomaspw.com.
Investment advisory services are provided by Ashton Thomas Private Wealth, LLC, and Ashton Thomas Securities, LLC, SEC-registered investment advisors. Securities are offered through Ashton Thomas Securities, LLC, a registered broker-dealer and member of FINRA SIPC.